guys, how have the trials been going?
They've been going. Not really sure where, but like they've been going.
Alright, well now that I've found a way to fix the non-functioning immune system in the mice, I think it's time we start human trials.
But didn't most of the mice die during the trial, even though only some of them survived? Like, just a suggestion, but shouldn't we fully test the trials on the mice more before testing on humans?
Yeah, I had the same concern, but I think I have the perfect solution. I'm going to create a human-monkey chimera, and that way it won't be human so we can test on it, but it'll react the same way a human would.
No, we can't create a human-animal chimera. That's against the law. I'll have to report you to a supervisor.
You can't start testing on real humans when the mice had so many problems when they weren't cured.
Alright, I guess I'll work out the kinks with the mice, and then we'll start human trials as soon as possible though. Honey, I'm home!
Hey, how was your day?
It was good! I think I found my next step for treating SCID.
Really? What is it?
I'm going to create a chimera to test my gene therapy on.
What's a chimera?
A chimera is an organism with genes from two different animals. In this case, I'm going to create a human-monkey cross.
A human-monkey cross? That seems wrong. What have your colleagues said about it, and how's that even possible?
Well, I'm going to make sure that the embryo has SCID by ensuring that it's homozygous recessive by taking your sperm and my eggs.
Whoa, what?
Yeah, and then once the embryo is developed into the blastocyst stage, then I'm going to merge them with monkey blastocysts.
That seems kinda shady, but if it's legal, I believe you. It is shady though.
So to treat SCID in the chimera, we're going to have to use the CRISPR system to edit the genes.
Okay, so what do we need to gather?
Okay, we need to gather the Cas9 protein, the guide RNA, the CRISPR system, and the replacement DNA.
Okay. Okay, I got that. What now?
Since the chimera is immune compromised, we're going to have to rebuild its immune system.
And so we would need to gather stem cells from the bone marrow, right?
Yes and no. One way would be to gather the hematopoietic stem cells from the bone marrow, and then we would use the CRISPR-Cas9 system to edit the genes by replacing the mutant gene with the correct gene.
Is there a less invasive way than extracting from the bone marrow?
I'm glad you asked. The less invasive way would be to take a skin biopsy from the chimera and then induce pluripotency to those skin cells. Those iPS cells would be able to differentiate.
Well, how would we get them to differentiate with the cells needed for the immune system?
Well, we would need to put them in the correct cell medium and then insert the correct growth factors. All right, we need to get started. We don't have much time to waste.
You're right. It looks like we have everything ready.
Do we have the skin sample from Georgie?
No, not that. I can go do that right now.
All right, be gentle.
Perfect. So now we're all set. I don't mean to overstep, but do you think that an autologous transplant is really the best option here?
Yeah, I see where you're coming from. It is hard to obtain true iPS cells, but I think it is the right option because there's no risk for immune system rejection.
Okay, but what about an allogenic transplant?
An allogenic transplant? That means we would have to find a donor who's willing to donate their cells, and that means paperwork and consent forms due to the common rule. Too many kids are dying from SCID, and I am not going to waste another minute.
Okay, you're right. Let's just start working. What's the first step?
Okay, well I have these retroviruses, and I'm in the process of deactivating them by deleting this gene so they can't reproduce. And then I'm going to add the transcription factors.
Well, do you want the skin biopsy?
Yeah, that would be great.
Okay.
Now I'm going to hand you this test tube, and you're going to put the retroviruses in the skin biopsy.
And then how long are we going to have to wait?
2 to 3 weeks, and then we'll see.
Well, hopefully these cells return to their pluripotent state.
I really hope so too. But the field has come a long way in IPS procedures. Hopefully luck is on our side. Looks like everything worked perfectly. Looks like we can continue on with our experiment.
Well, yes, thankfully it is, but I don't think we should jinx it.
Don't be so superstitious.
So here we've got the Cas9 protein, and we've got the CRISPR, and we just need...
The guiding RNA?
Yeah.
I'm working on that right now. We should have one that would match the mutant ADA gene.
It's kind of amazing how just one malfunctioning enzyme can cause such disastrous effects in a person.
Yeah. Okay, well I'm done. Now we can just insert the CRISPR-Cas9 system and that should do the trick. Here's the test tube.
Okay, sounds good. I'll go prepare the medium.
So here are the requirements for the medium. All we have to do is get these patient-specific iPS cells to differentiate into hematopoietic stem cells by following these instructions.
Okay, great.
Thanks, that's great. Now you can just transfer the iPS cells onto the new medium.
Okay, great. I'll do that right now.
Wow, looks like I missed out on a lot during vacation.
Yeah, you definitely did.
Hi, glad to have you back. So I just checked on the iPS cells and it looks like they're ready.
Oh, at last.
What exactly are you two doing?
Well, we're trying to find a cure for skin. We're gonna take these hematopoietic stem cells and inject them into this human monkey chimera to see if they'll treat the disease.
What kind of chimera?
Don't worry about it. Just help me hold him down.
Alright, you're the boss, let's do it.
And then we can walk to the chimera. Hold him down.
Hey Cameron.
Hey, what's up?
Pretty good. Last day on rotation for me. Anything exciting tonight?
It's quiet.
Too quiet. Did you see him?
I don't know, I haven't seen him.
I'm here. How goes it?
We have a list of tasks from Dr. Science for the night.
Okay, let's rumble.
Anybody want some tea? Yes. Okay, so prep plates for school visiting next week. Make sure we have enough C. elegans, clear lab stations, clean flasks and such, stock instrument cabinets, check glass for cracks, and monitor lab animals. Basically the usual.
Wait, what's that?
I'm not sure. Dr. Stephanie Science told me they're working on something important, so let's keep an eye on that thing. Probably don't want to stir things.
Cool.
What was that?
What?
I definitely heard something. I did not sign up for this. I'm out.
Oh my gosh, it's having a seizure.
Let's check it out.
We don't even know what it is. I guess we have to.
Hold it down.
Okay, it's calming down. We're good. What the hell happened?
I don't know, but we do need to get a CT scan. Call the hospital. Wait, no. Call the vet. No. Call the morgue. They have an old machine. We need to get this thing checked out. Let them know we're coming.
Yep, looks like tumors.
We don't even know what it is. Let's go check it out.
Wait, look, it has a wrist tag. 8932C, human chimpanzee chimera hematopoietic transplant. They don't mean chimera like chimera, right?
Hematopoietic transplant? Tumors? Chimera? What the hell is Dr. Science doing? Let's put this thing back into the cave. I don't understand. I thought that Dr. Science discussed the chimera with the others and was turned down. Does she not understand how unethical and dangerous this could be?
Well, at least it isn't moving now. Hopefully it stays that way until we can move it back into its cage. Wait, should it even be in a cage? Since it's part human?
We don't have time to discuss this right now. Our main problem for the moment is figuring out where these tumors came from. Any ideas?
They did use iPS in the lab?
It must have been a retrovirus.
Huh?
There are 4 common factors: OX4, SOX2, KL4, and CMyc. The deactivated retrovirus are reprogrammed with these genes, which will later be reverse transcribed. Sometimes, however, these retroviruses will randomly insert the DNA.
So
when DNA is inserted randomly, especially into the wrong locations where it may disrupt an exon, the genes can become mutants, and these mutants can often lead to malignant effects, sometimes in the form of tumors.
So you're saying that the retrovirus initially used to induce pluripotent stem cells from somatic cells is what caused the tumors in the chimera's brain?
Precisely. This can lead to aggression, which is what may have caused the chimera to act out in the first place. Of course, there are other factors that may have contributed to the aggression.
Like what?
Methylation of histones, other possible epigenetic effects, expression of aggression genes. There are quite a few. Do you want me to explain again?
No, I think I've got it. Histones and DNA methylation can sometimes cause genes to not be expressed, leading to high stress, anxiety, etc. We've seen examples in mice experiments before. And genes like MAOA for monoamine oxidase can sometimes also lead to aggression, depending on the levels of expression. This human warrior gene may have been inherited from the sperm or egg donor.
Good. It seems like you have an understanding of what may have caused this. My next concern is why it ended up acting so human-like.
And action.
There are 4 common factors: OX4, SOX2, KL4, and C-MYC. The deactivated retrovirus are reprogrammed with these genes, which will later be reverse transcribed. Sometimes, however, these retroviruses will randomly insert the DNA.
So
when DNA is inserted randomly, especially into the wrong locations where it may disrupt an exon, the genes can become mutants, and these mutants can often lead to malignant effects, sometimes in the form of tumors.
So you're saying that the retrovirus initially used to reprogram or maybe that's what caused the tumors as they grew uncontrollably. I mean, after all, we don't even know which cells Dr. Science chose for human DNA replacement.
That's an idea. But I'd say it's pretty unlikely. Hopefully we can ask her soon and get her to shed some light on this whole situation. Let's finally transport this thing.
Time to eat. No.
911, how may I direct your call? What? Sir, we need to keep these lines open for emergencies. Sir. Okay, please hang on. We're sending officers to your location.
We interrupt this broadcast for breaking news.
This Justin and genetically modified monsters rampaging the streets downtown.
The question is this thing even human? We have reports of it being half monkey and half man.
Is that even possible?
Well, that question is pretty existential in nature. What defines a human?
Some say that a human is defined by its potential, but some define a human by its ability to achieve.
That means that the life is very tricky to define. What do you think, Rylan?
I think that a human is an organism that is human genetically, but also conscious and cognizant.
Well, that's an imperfect definition because people who are comatose are not fully conscious.
See, this is what I mean by it's tricky to define humanity.
In any case, why does this thing even exist?
Science has been progressing in so many different directions.
Yes, folks, this was a result of an experimental procedure.
Debates are currently underway as to whether or not to outlaw this type of research.
This could cause concerns as this technology can also be used to help people with gene therapy.
More on this with specialist Dr. Stephanie Science and Dr. Dan Daniels.
Welcome back to the debate between Dr. Dan Daniels and Dr. Stephanie Science.
After the monster was accidentally created when scientists attempted to make a human-animal chimera, questions have arisen as to whether or not scientists should own the monster. Here are the various perspectives on this debate.
I do not think that the chimera should be owned. It is illegal and morally wrong to have ownership over a human being, and the chimera has human characteristics. It is half human.
I think that the monster can be owned because it doesn't even meet all of the requirements for being a person. First, it has no moral agency. It cannot even tell what's wrong and what's right. Second, it doesn't even have the potential to be a full human, whatever that means. For these reasons, because it's a human-animal chimera, not a human-animal chimera, just a human-animal chimera, it can be owned because it's not even a person. Case and point, thank you very much.
Well, this situation brings up the question of whether the human-animal chimera research should even be done, since it's part human. Ethics in human research requires that there's informed consent and lack of undue influence and lack of coercion.
As you can see, there are many different perspectives on this debate. In this ongoing debate, we will continue to think about ethics in research and create policies to clearly guide ethics in research.
More breaking news from the campus of UCLA. The famous monkey human chimera that has been rampaging LA the past couple of days has been sighted stealing bananas from B-plane. We have a report on the scene.
I'm here at UCLA campus where the chimera has recently been spotted in its utter chaos. A student originally saw the chimera trying to steal a handful of bananas out of B-plane, which is an action strictly prohibited by dining hall regulations. I am here with a student who saw everything. Please tell me about your encounter with the chimera.
Well, originally I thought it was a student, since a lot of people try to smuggle food out of the dining halls, but then I realized I saw this person before. I saw him in a news article online.
Has the monster been captured?
I don't think so. The workers at B-Play tried to stop him, but he was too fast.
Oh no!
This guy anyway.
I guess we take him back to the crazy scientist who made him?
I say we just kill him now. I mean, he's done nothing but hurt the city, and it looks like he's wearing a USC shirt.
Or we could just take him to jail.
But does the law even apply to this thing? Is it even
Well, you know what? He's still mostly human, so I think that if anyone should be punished, it's the scientist that created him.
Thank goodness the police got this monster off the streets. Like, look, it's in a cage and it's still freaking me out.
Yeah, well, now that we have control of it, what do we do with it?
Well, first we need to figure out what's genetically wrong with our monster, and then we can figure out how to fix it.
Guys, I found out what's wrong with Georgie. He has a genetic mutation that can't encode for human features.
So what should we do?
Well, what if we use somatic cell nuclear transfer to clone Georgie, and then we can have another subject to test on?
But where are we gonna get an egg donor since there's no female monster?
Yeah, and it's also unethical to use a human egg cell to clone a monster.
So if we can't clone the monster, what if we cured the monster of his skid with induced pluripotent stem cells? We could take his somatic cells and induce pluripotency to them, and that way it could differentiate into any type of tissue.
That's definitely more plausible than cloning cells, but introducing more pluripotent stem cells runs the risk of giving the monster cancer. These cells can divide rapidly in a certain area of the body and metastasize and invade other parts of the body.
Okay, so we can throw that idea out. What else can we do?
Well, we could use a retrovirus and insert our gene of interest and cure the monster.
That's crazy enough to just might work.
As the scientists continued their research on the monster in their lab, the public has expressed its outrage regarding the monster and the research that has created it. People outside are protesting the research, and we really must ask ourselves, is the scientists' research really benefiting mankind? Or are the scientists playing God and trying to create their own fame?
I believe this being has the right to live. He deserves to be happy, and we as human beings owe him this after all he's been through.
We have this moral obligation to protect this being from this scientific torture, man.
Kill it, kill it, kill it, kill it, kill it, kill it. It's an abomination.
We should send that thing to space. Let it find its own way, then we won't have to deal with it.
These scientists should rot in jail for what they've done.
Don't worry. You can be happy. You'll never have to go through those painful experiments ever again. What those scientists did to you was totally wrong.
It has now been a year since the incident involving a mad scientist's horrible experiment occurred here at UCLA. Where is it? Is it dead? Is it creating chaos? Or maybe were we so wrong all along and it was just an elaborate hoax made by USC to hinder UCLA's greatness? Only time will tell.
As you have heard from this tale, scientists try hard to combat disease, death, and uncertainty. Professionals in the medical field are continuously striving to make advancements, trying to cure the incurable and prolong the human lifespan. SCID may be one of the most heartbreaking diseases. It affects the youngest members of our society who are constantly threatened by bacteria, germs, and minor illnesses that are usually harmless to those with healthy immune systems but could be fatal for people with SCID. The ambitious scientists portrayed in this story blurred the lines between legal and illegal and brought to light an issue that plays a major role in the professional lives of researchers. The necessity of progress and the need to maintain certain ethical boundaries are constantly at odds. There are currently a few effective treatments for SCID. The three ways we know to treat SCID today are through bone marrow transplant, enzyme replacement therapy, and gene therapy. The only known cure for SCID is bone marrow transplant. This transplant has proven to be effective, however, it does come with some disadvantages. The process of donating bone marrow is somewhat long and requires surgery, which always comes with some risk. Also, because the bone marrow is coming from someone other than the patient, there's always a chance that the patient's body will reject the transplant. This is why researchers are exploring gene therapy and enzyme replacement therapy. While these treatments have had some success, more research is required before they can be used to cure the disease in all patients. Even though there is still research to be done that could benefit SCID patients, the creation of a human-animal chimera for the purpose of research is not permissible in today's society. This is the perfect example that pits ethics against scientific advancement. Despite facing ethical boundaries, researchers continue to push through current medical limitations and perform cutting-edge procedures in order to eradicate this calamitous disease.